So, Chapter 10, Part 2, we're actually going to begin talking about the interactions between the drugs and the microorganism. Basically, what we're going to focus on is how the drugs work. As we go through this, do not attempt to memorize drugs. That is not the goal of this course. Instead, for this chapter, what I would like for you to be able to do is identify a mechanism of action if it's described to you. So be able to tell me how something works or why it falls into a specific category as far as modes of action are concerned. Now, the big goal of antimicrobial drugs is to kill or inhibit some pathway, some product, some action, or synthesis of a molecule in a microorganism, but not invertebrate cells. In other words, we're trying to maintain that selective toxicity that we were talking about, the us versus them. Okay, we want it to work on the bacteria, but not work on us. Okay, so usually what we see is disruption of some type of cell process. In viruses, we inhibit replication, interfering with enzymes, or it's possible to destroy structures that are already formed within the cell. It just depends upon the drug itself that you're actually using. So, again, that big goal is selective toxicity. But not all drugs we use will have excellent selective toxicity. Drugs that are particularly toxic to humans will act on structures that are common to both the infective agent and the host cell. So things that the eukaryotes and the prokaryotes have. So if you can think of a structure common to both bacteria and animal cells, see if you can name one. I'll give you a minute. Hopefully you were thinking cell membrane. Okay. Not a lot of selective toxicity in the cell membrane. Phospholipid bilayer is phospholipid bilayer. The cell membranes for bacterial cells and for animal cells pretty much match one another. There's not a whole lot of difference there. So when we do see drugs being particularly toxic, it usually has something to do with the fact that we're trying to target something that exists in both the eukaryotic and the prokaryotic cell. It's one of the reasons why we don't have a lot of medications to treat eukaryotic microorganisms like protozoa and helminths. They're out there. We do have treatments. The unfortunate part is that a lot of times people will get sick because of the treatment. The treatments themselves have a tendency to work on parts of eukaryotic cells, and that's what our cells are, so they affect us as well. They have very poor selective toxicity. Now, drugs with excellent selective toxicity find structures that are common in bacterial cells but not found in animal cells. So can you think of one of those, or maybe some of those? Probably the most recognizable is going to be the cell wall. Okay? Cell wall, bacteria have it, our cells do not. Okay? It's probably going to be the best mechanism of selective toxicity because destroying the cell wall is going to be really bad for the organism, but should not affect us because our cells do not have cell walls. So, we're going to talk about 5 mechanisms of drug action, specific mechanisms. So, targets, sort of big things to remember. Okay. Inhibition of cell wall synthesis. Okay. Nucleic acid structure and function. Okay. Inhibition of protein synthesis. Interference with cell membrane structure and function, okay, and inhibition of folic acid synthesis. I will be the first to tell you that we are probably not going to call this. When you see it on the test, this will be termed action as an antimetabolite. Okay? Inhibition of folic acid synthesis is probably the best example of action as an antimetabolite. Okay? So if you're a particularly visual person, this is an image out of your book that shows you drugs and where they target within the bacterial cell. So cell wall inhibitors, cell membrane, the DNA or the RNA, protein synthesis, folic acid synthesis. Okay. And then list of drugs if you're still working on your mechanisms of action report and the MOA. Okay. Now, again, I'm not asking you to memorize these. Do not go through these and start memorizing. That's very general uses and basic characteristics. But you will note that these are divided into those that target the cell wall, those that target protein synthesis, okay, those that target folic acid synthesis, DNA and RNA, and then the cell membrane. Okay. So, let's talk about the bacterial cell wall as a target. Okay. So that first mechanism of action, cell wall synthesis. Okay. Okay. Probably the most appealing target for antibiotics. Why is that? Well, the answer is selective toxicity. Okay. There is no cell wall in human cells. Okay. In other words, we're not going to have cell walls. That makes it easy to use and have a decent amount of selective toxicity so that they don't work on us. Okay. Now, things about the cell wall. If you remember, the new cell wall is continuously built during the log phase of growth. This is when we are making new cells. And hopefully you also remember that cell walls are made up of two sugars, NAG and NAM, that when put together make peptidoglycan. Okay. They are cross-linked through the activity of two enzymes. I will be happy to go ahead and tell you that yes, you are going to want to remember the names of these enzymes. And remember that they are all about building cell walls. Okay? They're linking NAGs and NAMs together to make peptidoglycan. Okay? So without these enzymes, cell wall synthesis stops. Okay? Now, a lot of antibiotics actually inhibit the activity of transglycosylase and transpeptidase enzymes. That makes them targets of antibiotic molecules. And when they are targeted, they result in improper cell wall cross-linking. So if the cell wall is not built correctly, the cell wall will not function. And if we remember, okay, cell walls do the two S's. Okay. Shape, maintaining the cell shape, and stability under osmotic pressure, keeping the cell from rupturing. Okay. Now, a great example of cell wall synthesis inhibitors are the penicillins. Okay. The most important part of penicillin molecules, and that's all of the penicillins. These are a class of antibiotics, so the penicillins, okay, not just penicillin itself. The most important part of penicillin antibiotics is this structure right here, this beta-lactam ring. Okay. What it does is actually bind the penicillin molecule to the enzymes, okay, the transglycosylase to the transpeptidase. It will bind this molecule to these enzymes, preventing the enzymes from functioning, making it difficult to construct new cell wall. Okay, so the beta-lactam ring is the functional part of a penicillin. So these interfere with producing new cell wall. Because of that, penicillins are most effective during the log phase of growth, because the log phase is when cells will be actively trying to link NAGs and NAMs to one another. We're actively trying to make new peptidoglycan to make new cell wall. Now, any mutation in bacterial genes for the transglycosylase or transpeptidase proteins actually changes the shape of the enzyme slightly, allowing for them to be resistant to things like penicillins. So next up, the cell membrane. Okay. Now, cell membrane, if we remember, is going to be the least selectively toxic. Okay. So it does not allow for selective toxicity because the bacteria cell membrane is very, very similar to the eukaryotic cell membrane. Another reminder in here, cell membrane, plasma membrane, cytoplasmic membrane, all of these terms are synonyms. Okay? They all mean the exact same thing. Do not get thrown off by terminology if one sentence says cell membrane and the other says plasma membrane. Okay? Now, these are really good targets for antibiotics because there are a lot of important physiological functions that happen at the plasma membrane. If you remember, okay, the cell membrane does the two R's. Okay. Regulation and reactions. We see membrane-dependent reactions like the electron transport chain happening at the cell membrane. And the cell membrane is responsible for regulating what comes into and out of the cell. Okay. So disrupting the cell membrane is going to be very, very bad for the cell. Our example, polymyxins. These act as detergents, so basically they work like surfactants. And if you remember, surfactant molecules looked almost exactly like phospholipids that were used to make cell membranes. Okay? And if you remember from the last chapter, these sort of wiggle in between the phospholipids, creating little holes in the cell membrane, basically disrupting the regulatory ability and pretty much all of the abilities of the cell membrane. Okay? So cell membrane kind of has holes poked in it by the surfactants. Cytoplasm is lost, cell substances are lost. Basically everything that makes the cell a cell is sort of lost to the extracellular fluid. Okay? Cell membrane inhibitors are actually quite effective against gram-negative bacteria, mainly because the outer membrane is high in phospholipids. That outer membrane, or the LPS, the lipopolysaccharide layer, looks a lot like the cell membrane. Lots of phospholipids. The surfactants will work on the phospholipids there just like they would on the cell membrane. So these are particularly good at affecting gram-negative organisms. If we think back to the last mechanism of action with the cell wall, hopefully you also understand that because antibiotics like penicillin affect the cell wall and the cross-linking of NAG and NAM, they're particularly effective against gram-positive organisms. Okay? The gram-positive organisms are going to have a lot of NAG and NAM, lots of peptidoglycan within the cell wall. The gram-negatives, less peptidoglycan, and the peptidoglycan will actually be protected by the outer membrane. So those types of antibiotics are usually slightly less effective on the gram-negatives. So, synthesis of bacterial proteins. Now, this is protein synthesis specifically. So, think translation. Okay. If you're trying to mess up translation, probably the best bet is going to be to go for the ribosomes. Okay. So our targets here are going to be the ribosomes. Now, is this an example of selective toxicity? Are ribosomes in bacteria any different than ribosomes in humans? Well, the answer is yes. If you remember, eukaryotic ribosomes, 40S, 60S ribosomal subunits together make an 80S ribosome. And in the prokaryotes, 30S small subunit, 50S large subunit, and together they make a 70S ribosome. Okay? So, can we get selective toxicity? Well, sort of. Okay? One thing we have to take into account is that eukaryotic cells have mitochondria. Okay? And if you remember all the way back to chapter 1, mitochondria were some pretty good evidence of what we called endosymbiotic theory, stating that prokaryotes gave way to eukaryotes, or sort of helped with the creation of eukaryotes. And part of our evidence was the fact that mitochondria look a lot like bacteria and have a lot of bacterial features. Specifically, that mitochondria actually have 70S ribosomes. So because mitochondria actually have the same ribosomes that we find in prokaryotic cells, if we give antibiotics that affect 70S ribosomes or parts of the 70S ribosome, like the 30S subunit or the 50S subunit, in excessive amounts, it can actually inhibit the function of mitochondrial ribosomes. Okay. So a couple of examples of this. Okay. So we're going to talk about a few antibiotics that specifically deal with the ribosome or interfering with protein synthesis. Now, if you remember, okay, when we talk about the ribosome, there's a 30S small subunit, okay, and the 50S large subunit, and there's 3 spots within the ribosome, okay. And if you're going to kind of pull some information from there, if you remember, we A site, P site, E site, okay, and we ape our way through the ribosome. The 50S subunit and the 30S subunit actually have different jobs. So we see peptide bonds made here. So we're attaching amino acids to one another. On the 50S subunit, the 30S subunit, okay, helps feed our mRNA, okay, through the ribosome. Nice and straight so that it's readable and not crinkled up. Okay. Aminoglycosides like streptomycin actually insert themselves in the 30S ribosome, sort of acting like a speed bump so that mRNA is misread. If the mRNA is not read correctly, remember we had to read those codons in groups of 3s. If we can't read those codons and the groups of 3s don't stay intact, we get abnormal proteins made. Abnormal proteins, okay, are basically non-functional proteins. They're not going to work. Okay? Now, because all bacteria will have ribosomes, this is going to be a broad spectrum antibiotic. Okay? Has to be. All bacteria have ribosomes. Okay? Tetracyclines, they block the docking site of tRNAs at the A site. So tetracyclines will actually set themselves down, okay, right on the A site and prevent tRNAs from bringing in new amino acids. Okay. Well, when that happens, right, new tRNAs cannot come into the ribosome. No new tRNAs means no dropping off amino acids, means no proteins. No proteins, the cell is pretty much dead. You can't make new enzymes, can't make new structures. It definitely cannot make any new cells. Okay? This is bacteriostatic. It's not going to outright kill the cell. If this were to kill the cell, it would be bactericidal. The enzymes that are already made and the proteins that are already made will still be left intact. This just prevents production of new proteins. Okay. Tetracycline isn't used anymore. Long use of tetracycline has led to a lot of resistant organisms. Again, broad spectrum works against gram positives and gram negatives. Okay? And that, one more time, is because all of these have ribosomes. Okay? Working on the 50S subunit, clindamycin. Usually we see this being more active with anaerobic organisms and protozoa. It works by interfering with peptide elongation. So the joining of amino acids is disrupted. A major disadvantage to clindamycin use is that it's commonly associated with antibiotic associated diarrhea, including Clostridium difficile, or what we call C. diff. Okay. Mainly because this one does a pretty good job of wiping out those normal intestinal flora, normal intestinal bacteria. Okay. Chloramphenicol, also not really prescribed anymore, sort of being reserved in use for extreme cases. Binds to the 50S ribosomal subunit and prevents the addition of amino acids to the growing peptide chain. So it works kind of similarly to the clindamycin. Erythromycin specifically blocks approach to the exit tunnel. So that exit tunnel where we saw those tRNAs leaving, erythromycin prevents those tRNAs and the amino acids themselves, the joint polypeptide chain, from leaving. Therefore, it prevents long strands of amino acids from being built. Okay, so we have issues joining amino acids to one another. That's making a protein. All of this is inhibiting protein synthesis. Now, nucleic acids, we're talking about inhibiting DNA and RNA. Now, DNA and RNA are universal components. In other words, DNA and RNA are the same in all types of organisms. Therefore, you can't have any selective toxicity if you actually target DNA and RNA. Okay? So we can't break DNA that's already there or break RNA that's already there. That would be a disaster because anything that broke bacterial DNA would also break human DNA. Okay? But there are some differences between bacterial and animal enzymes used to synthesize nucleic acids. So what we're going to inhibit is nucleic acid synthesis. That allows for some selective toxicity. Okay? Those slight differences in the enzymes. Now, these include two families of synthetic compounds. In other words, these are man-made antibiotics, specifically the rifamycins and the quinolones. Let's talk a little bit more about each. Okay. In the rifamycin family, the only one I'm aware of that's actually on the market in the U.S. is a drug called rifampin. Okay. And rifampin works by binding to bacterial RNA polymerase. And if you remember, RNA polymerase is responsible for making RNA. Okay? It binds to those promoter sites on the genes and turns DNA into RNA. So this is responsible for the process of transcription. Okay? Now, rifampin binding to bacterial RNA polymerase means that no RNA is synthesized. Well, we know that if you do not make RNA, you will also not make protein. So not making RNA is basically inhibiting the cell from reproducing. Okay? Now, I will tell you rifampin is only used in combination therapy. We found that the resistance develops extremely rapidly when it comes to rifampin, so it's usually used with another drug. Most commonly, we see it put together with a protein synthesis inhibitor, so that you're basically disrupting the synthesis of proteins from two ways. Okay. The other nucleic acid synthesis inhibitor is nalidixic acid, the active ingredient in the quinolones. They target bacterial topoisomerases, and if you remember, topoisomerase was responsible for taking that supercoiled bacterial DNA and unraveling it so that it could be unzipped by helicase and used during DNA replication. Now, because bacterial topoisomerases are different from eukaryotic topoisomerases, they make a really good target. They give selective toxicity to the process. And when they're targeted by nalidixic acid, this blocks the movement of the replication fork. If the replication fork doesn't move, replication doesn't happen. Okay. And new DNA is not made. No new DNA means that you cannot make a new cell. Okay. So this again inhibits bacterial growth. Last up, action as an antimetabolite, or the folic acid synthesis pathway, very specifically inhibiting the folic acid synthesis pathway. In order to understand the antimetabolite part of it, you first sort of need to know what a metabolite is. And a metabolite is a metabolic intermediate, basically something used in a metabolic process. An antimetabolite prevents you from using a metabolite. Okay? So an antimetabolite basically prevents a metabolic process from being carried all the way through. Okay? Now, there are two targets for inhibiting bacterial growth: the production of nucleic acid precursors, or metabolic pathways that happen at the plasma membrane. Now, the one most commonly used and the one we're going to focus on is the folic acid synthesis pathway. Okay? Action against the folic acid pathway has good selective toxicity because we don't synthesize folic acid. Humans do not make folic acid. Instead, humans obtain folic acid through their diets. Orange juice, leafy greens like spinach. We ingest our folic acid. It comes from the outside environment. Bacteria, or some bacteria, not all, but some bacteria choose to actually synthesize their own folic acid. Okay? So if we attack the folic acid synthesis pathway, it's selectively toxic because we don't have a folic acid synthesis pathway. Okay? So this is what the folic acid synthesis pathway looks like. You can see it here. The intermediate para-aminobenzoic acid, PABA, you can see it right here. Okay. Normal enzymatic action, so ARO. There's an enzyme here. An enzyme helps convert PABA to folic acid. Folic acid, through the action of several other enzymes, is converted to nucleotides. Those nucleotides are then converted to nucleic acids. Okay? Things like DNA, RNA, ATP in here. These are extremely, extremely important. We need these. Okay? So not having folic acid because you can't make it is going to be really, really dangerous for the cell. Sulfa drugs inhibit the folic acid synthesis pathway. They act as an anti-metabolite. And basically sulfa drugs prevent this reaction right here. Okay? Well, if you can't make any folic acid, there's never any production of nucleotides, so these don't happen. And if there aren't any nucleotides, there are no nucleic acids, so this piece doesn't happen. Okay? Inhibiting one part of the chain automatically inhibits the rest of the chain. Right? The pathway itself stops, and this becomes a lethal event. So how does this happen? You can see the sulfa drug inhibiting the pathway here. Right? How does this happen? Well, This molecule right here is PABA. Okay? And this little orange jelly bean you're seeing is the enzyme that's responsible for turning PABA into folic acid. Okay? So this is the enzyme that turns the PABA into folic acid. It helps with the conversion. So this is the enzyme that's being used in this process. Okay? So if you look, the reason enzymes work, and we know this, we remember this from earlier in the semester, has a lot to do with the shape of the active site. The substrate fits into the active site, and then that is what helps it be turned into a product. Okay? Well, so if this is PABA, this is a sulfa drug. If you notice anything about them, it's that the sulfa drug looks a whole lot like the PABA. In fact, it has the exact same structure that allows the PABA to fit into the enzyme. The only difference is sulfa drugs cannot be turned into folic acid. So what happens is the sulfa drug is erroneously incorporated into the enzyme, so it's put in by mistake, okay? But the sulfa drug is never converted to folic acid. This shuts down the folic acid synthesis pathway, okay, and prevents the cell from making any nucleic acids. Okay? If you remember from chapter 6, anybody remember what type of inhibition this is? Okay. So you said competitive inhibition, you were right. Okay. The sulfa drug outcompetes the PABA. So, this is the end of part 2, mechanisms of action. Part 3 involves resistance. So come back for part 3 next, and we'll talk about resistance to antibiotics and the 5 mechanisms of action of resistance.